Hi, and thanks for coming to see the Beneath the Surface Day 7 materials that we have. First, in class, everybody got out water quality packets and answered the following warm-up questions. So when we want to think about why water clarity is important, it is important because clear water allows the sunlight to reach our plants, which will allow them to do photosynthesis, and it allows us to have a higher level of increased dissolved oxygen. So clear water leads to more oxygen in the water. When we want to think about causes of nitrites increasing, typically we'll have increased nitrite levels if we have more animal waste or if we don't have enough of our good bacteria to help combat the nitrites or change those nitrites into a better form of nitrogen. Also, if we don't have enough plants, we might have high levels of nitrites. If we think about a deer population, what could be an example of an abiotic limiting factor for a deer population? Remember, abiotic means it's not living. So a limiting factor that is abiotic might be if we had forest fires or if we didn't have enough water. And if we think about what helps turn our nitrites into something that is not harmful, as we were talking about levels of nitrites increasing, that happens if we don't have enough of our good bacteria. So good bacteria do help turn nitrites into a form of nitrogen that we can use. And finally, what is the water quality indicator or the water quality test that we use to find out how acidic water is? That's our pH test. All right, so these questions should help you be prepared for our test that's going to happen next class. Also, there are extra materials in Schoology that you can find in the Beneath the Surface Schoology folder. There's a new folder that is extra resources. There's at the very least a quizzes in there and a Kahoot. But remember, you can always go back through all of your exit tickets to make sure that you know what's happening. Okay, true or false, Mrs. Thomas will take points off if you don't write your first and last names on your papers. That is true, so please be sure to write not just your last initial, but your whole last name. All right, our test next class is going to cover ecosystems. Remember, we want to know about biotic factors and abiotic factors of ecosystems and how they interact. Also, limiting factors. Remember, limiting factors, that's a factor that's going to slow or reduce the number of a species or a population that can continue to grow. So when we talked about bunnies and foxes, the foxes were eating some of the baby bunnies, and that's the reason we were not overrun with bunnies. So foxes in that case would be predators. Those would be biotic limiting factors. Abiotic limiting factors might be shelter or perhaps water or temperature or salinity. There's lots of things that are abiotic also that would possibly limit the growth of a population. All right, remember that we have two different kinds of observations we talk about. We have qualitative observations. Those usually have adjectives, fluffy or soft or green. So if we are using our senses to make an observation, that is going to be a qualitative observation. However, quantitative observations are things that we have quantified or counted or measured, and so we will generally have some sort of number and unit. So we might have 5 kilograms or we might have 3 milliliters or something for a quantitative observation. And then, once we have observations, we have background knowledge that we use to make inferences. So an inference is a logical conclusion that you've come to that is using observations and some of your background knowledge to help try to explain something. Okay, finally, make sure that you understand the water quality test that we're doing in class, and you understand why we are testing for those things and what we might do if our levels are not in a range that is comfortable for our ecosystem. All right, our warm-up question was, would you rather scuba dive or go on a spacewalk? And we discussed those a lot. So continuing to review for our upcoming tests, what are 2 ways that we can get more oxygen into the water? Please remember that if we put more plants in the water, they're going to perform more photosynthesis, and so they will add oxygen to the water. Also, if our water is moving water has more oxygen in it. And cold water can have a higher dissolved oxygen than warm water. All right. How will oxygen get put into our ecosystems in the classroom? Well, we are adding some things. We have just added some things to our ecosystems. This image right here is a plant that you can see is performing photosynthesis. You can actually see oxygen bubbles on the outside of that plant. One reason for nitrites to increase is fish waste. Okay. Also, if we don't have enough of our good bacteria, that can also increase our nitrite level. Okay, so if we imagine that we have students in the classroom who are looking at the results of our tests, which is what we always want to do, we want to look at the results and think about what does that mean to us. So if this person in green says the dissolved oxygen is at 3.2, what do I do? This guy says the nitrites are at 2. Is everything going to die? Because that's outside of our normal range as well. And finally, the pH is 5.1. How do we fix it? That's our big question. How are we going to fix our ecosystem so that we can get our water quality back to normal? So if you look in your packet, you'll find this page. And we can talk about water temperature when it's too high or too low. With dissolved oxygen, we don't care if it's too high, but we do worry if the dissolved oxygen gets too low. pH, we don't want it to be too basic or too acidic. Remember, if it's basic, the numbers are higher than 7. If it's acidic, the numbers are lower than 7. And then for nitrites, we do worry about nitrites getting too high. We don't really worry about nitrites getting too low. Finally, water clarity. The clearer the water, the better photosynthesis we have and the better dissolved oxygen levels. So what do we do if our water is too murky? All right, so if the water temperature is too high, we're going to lower room temperature. That's a way to make our ecosystem be cooler. Or we could move our ecosystem out from under a sun spot. If it's in a sunny window area, we might want to move it. We could add colder water, but that'll probably only be a temporary fix. If the temperature's too low, we can raise the room temperature. Or we can move that ecosystem into a spot in the room where it might get more sun and get warmer. When we think about dissolved oxygen, again, we're not worried about dissolved oxygen being too high, but if it's too low and we need to make it be higher, we can add plants. We could lower the room temperature, and maybe we would use a bubbler to make that water have some movement. Okay, I'm going to back up in our slides here a little bit. So for pH, if something is too basic, the pH number is too high and we need to make it more acidic, we might want to put in more fish, because the more fish breathe in water, the more acidic that water becomes. If our pH is too low, we might add plants, or we might try to reduce the amount of acid rain that a body of water is exposed to. It's going to depend. Obviously, we don't have acid rain happening in our classroom. If our nitrites are too high, we're going to try to add in some of our good bacteria, or maybe take some fish out so that we have less animal waste. We're not worried about nitrites being too low, so we're not going to do anything about that. And if our water is too murky, if that number for the water clarity is too high, we can take out some of the water and replace it with cleaner, clearer water that has less sediment in it. All right, so let's get through some of these slides that we looked at. You've really covered all of the things that we need to cover about our ecosystems, and we just need to keep practicing and make sure we understand the organisms that we have added to our ecosystems and what they are and what they can do for the ecosystem. All right, so what did we add? Right now, notice those guys are green. They do have these long spindly things coming out. Here's an image of our Chlamydomonas. Okay, so our Chlamydomonas has flagella. okay, 1 flagellum, 2 flagella, that help them move. They can actually move around in our aquatic system. Oops, I clicked the wrong button, sorry. Okay, so because of these flagella, and if we were able to see these in water, we would actually be able to see them moving. So they are a form of algae. They are green. They are able to do some photosynthesis. So you can see they even have some chloroplasts. Okay. All right. This is another thing that we have put in the ecosystem. This is called Spirogyra. It's a second type of algae. So under the microscope, you can see this is kind of what the structure looks like. If you're reaching into a pond that has a lot of Spirogyra, you can see it's kind of wispy, long strands. So our Spirogyra, we have a much smaller amount that went into each of our ecosystems, but it is also green, and it's kind of this green, wispy, strandy stuff. It is also green. So any guesses about how it gets its energy? Yep, it gets its energy from the sun, it performs photosynthesis, it creates its own food using sunlight. So the two algae that we've put in, they are producers. A producer, just as a reminder, is an organism that makes its own food using sunlight or through photosynthesis, right? So autotroph. The other word that we might talk about is a consumer. A consumer is an organism that eats producers or other consumers. Consumers cannot make their own food. All right, so why are we adding producers before consumers? Well, if we put consumers in there without any producers, they wouldn't really have food, especially the low-level consumers that only eat producers. So we're going to put in our producers first, hope that they get established, and then eventually we'll be adding consumers. All right, we added a third thing. This is called Daphnia. So its common name is a water flea. Daphnia is actually the genus. So this is the Daphnia pulex. Pulex would be the species, genus, species, right? And so we're looking, this is kind of an enlarged petri dish where you can see these Daphnia swimming around. This is very enlarged, probably through a microscope, so you can see that it has some organelles and things happening inside of it. And if you look at the diagram, it has a mouth. So if I asked you whether it's a producer or a consumer, your hint would be that it has a mouth. It has a mouth because it's going to eat things. So this is a consumer. This consumer likes to eat things in addition to lots of things, I guess. It would like to eat Spirogyra. It would like to eat Chlamydomonas. So it'll nosh on the algae that we have in our ecosystem. All right, this is a consumer. So now, I've asked everyone to please go to your data table. In your packet, we did day 0. That was our baseline data. So now we're going to work on day 1. And in the classroom, we talked about our room temperature. This is day 1. Day 1 for some of the classes was October 3rd, and some classes it was October 4th. So everybody has the same room temperature listing in this white box for our classroom for the class period that you are in. Everybody is in the same room with the same room temperature. But you do need to have whoever is the water quality person measure that water temperature and put that number in the gray box. So we have a white box and a gray box just to differentiate. The white box is for room temperature and the gray box is for water temperature. All right, so remember we're testing for dissolved oxygen levels and pH and nitrite and water clarity too, in addition to temperature. So take a look at your packet so that you know which test you are doing today. If you missed class, you want to make sure that you get the data from all 4 of these tests from your table mates, but you might want to practice the test that you missed doing as well. So you can make an arrangement to come during Falcon Focus to do that. All right, so these are the jobs for today, October 3 and October 4. And remember, when you go to your station, you always want to have your goggles on. Okay, at the end of each testing set, we want to just make sure that we haven't depleted too much water, because when you take your water samples out, you do not return them to the ecosystem. So we just want to make sure the water levels don't get too low. Make sure that those job cards are returned to the cup or the basket. And so that's just also a reminder that when you're getting up to do things like dissolved oxygen or pH, you want to have the instruction cards right in front of you. Okay, and then when you go back to your desk after you have finished your whole sample, you want to make sure first that you've put your sample cups and items back in their proper locations on the counters, but then you want to go ahead and put your goggles in your own baggies with your name, and then your baggie goes into your table's bigger baggie. Okay, once you're at your desk, or your table rather, and you have recorded your own data, you want to make sure that you get data from all of the people at your table, and you want to fill in all of these boxes. Okay, all of these boxes should be filled in for day 1. So we had about 12 minutes to complete that in class. And then students filled in their information so that each table had all of these boxes filled. And then you want to think about today's quantitative data. What changed? So please, if anything changed, write something like pH went up, or dissolved oxygen went down, or there were no changes. Just make sure you write something in this box. If there were changes, make some inferences about what you think might have caused your changes. Okay, if there weren't any changes, why do you think that was? All right, so with your group, you're completing those last 2 boxes. Oops, and we had 2 minutes to do that. Okay, we packed a lot of stuff into this class. So in Schoology, you will see a BTS Day 7 wrap-up/review slides. So you're going to get into those slides and you're going to learn some things and remind yourself about some things so that you're practicing for your test. So let me pull those slides up and just make some quick comments on that. Okay, so you're going to look in Schoology. It's called BTS Day 7 Wrap-Up and Review Slides. You have a link to get into those slides. And so you're going to go through these slides and you're going to do your very best to, for example, find the definition for biotic on the right. Biotic is something that is living or was once living. And you're going to drag that over so that biotic word is pointing to biotic definition. I'm not actually going to move that. I'm going to leave it where it is. Okay, go through all of these slides. Here, you're going to move different words from the left into the T chart that makes the most sense. And so you can see that you're just going to go through each one of these slides and interact with the slides just a little bit. Okay, you're putting some definitions and some observations and things in different locations on the slides. Then you'll submit these slides when you're done. Okay, try to be clear with your answers. You can be brief, but definitely be clear. Okay, I'm going to keep on scrolling down. Anytime there's a question, you want to try to answer it clearly. And so we have lots of questions. And then the very last slide says your job is to sum up what we have learned this unit with 6 images and an appropriate caption. So an example might be that you've decided that running water tends to have higher levels of dissolved oxygen. So I might Google in another tab running water images. And if I find a nice running water image, I can cut and paste the image here. And my caption might say running water has higher levels of dissolved oxygen. So you get to pick 6 different things based on anything from your slides or your notes from this beneath the surface set of information, and put a caption, and then you get to submit these slides, these review slides. All right, so back to our actual slide deck. Again, you're going to go through that wrap up and review slides set and go ahead and submit those. That is an assignment to submit. And then you can try to do the Beneath the Surface Day 7 exit ticket. You get 2 attempts for that, and you can use your notes, of course. And you should also try this review for test, practice test. That is also in Schoology. So you'll see we have several things in Schoology for you to do. This page is talking about two different things. Complete the BTS Day 7 exit ticket. Also try the review for test practice test. We do have a test next class. Okay, so again, exit ticket, you get 2 tries, and that review for test, you get 2 tries too. All right, finally, the last page in your packet, or it may not be the very last page, look for the page in the packet that says date added and organism's common name. So this page is the place where we get to write down what we added and when we added it. So remember today we added Chlamydomonas, that's one thing. We added Spirogyra, and we added Daphnia. So you want to have 3 different boxes filled in for the things that we've added. And then you need to add in the scientific name, the domain, the kingdom. Remember, scientific name is genus and species. Then you want to talk about how it gets its energy. Is it a producer or a consumer? If it's a consumer, what are some of the common prey? What might it eat? And then a quick drawing. Okay, so you can certainly Google how to draw something like Chlamydomonas or Spirogyra or Daphnia. But I'm going to show you where you're going to find this information. So you're going to go into today's folder and click on the slide deck that says Beneath the Surface Day 7 Organism Details, because that's where you're going to get details for these organisms. Please make sure you read through all the directions. If you see a box that says click here, you should, click there. And then if it opens something for you, go ahead and read it. So, for example, if you click on a box that says, to learn more about the kingdom and domain, click here, you'll then need to read the information that's on that next slide. And I promise, kingdom is in there, and you'll just have to kind of read through the words. So definitely, if you see words, go ahead and read them. When you have a Wikipedia page, you might be able to get by with just the first paragraph or 2. You probably don't need every single word from the Wikipedia page. In any case, we did add 3 organisms, so you are going to record that you have added 3 organisms and get as much of that information as you can. All right. So once you've done that, you've gotten some information about the 3 organisms we added. There is a page. Oh, goodness. Sorry. Hold on. All right. I know it wasn't a pause for you. There was a brief pause for me. So I'm trying to get myself back to what I was saying. You're finding a page in your packet that says Aquatic Ecosystem Daily Reflections. So day 1 for us was either October 3 or October 4. And so we're going to go ahead and put the date. And you're going to think about this question. What is a way that 2 of the 3 organisms you added might interact with each other? So remember, we added 2 algae, that was Chlamydomonas and Spirogyra, and we added Daphnia. So how might those organisms interact with each other? Just go ahead and put down some brief reflections so that you're thinking about why we are adding things to our ecosystems. All right, and then make sure you study for your test. Go through all your previous exit tickets so that you are ready to do well on the upcoming test. Thanks again for watching.